So, hello ma'am, good day ma'am, good day everyone. Today I will show you how to transfer bacteria from one petri dish to another. So first we have to sanitize the area with isopropyl alcohol. The alcohol lamp is used to burn and kill microorganisms. Then transfer the alcohol lamp into laminar box. The inoculating loop and the inoculating needle should be kept in isopropyl alcohol to keep it sterile. The, the inoculate loop needle needs to be heated over the alcohol lamp at 45 degrees Celsius, and it should be done until it's red hot. Then next step is to take petri dish, which is here. The next step is to take petri dish which contains bacterial culture and ensure that the inoculating loop is not too hot, and check the temperature of it. You just have to tap on the edge of the petri dish, into four imaginary quadrant, one, two, three, four, into four imaginary, and then streak the bacteria in four quadrants. Then you have to sterilize the 1 liter over the alcohol lamp. Once it's red hot, you have to wait to cool it down. Then from the first quadrant, streak it to second quadrant. You have to repeat the process and sterilize and streaking your bacteria until all the four quadrants are covered. Then next step is to seal your petri dish. And keep it upside down at the room temperature. So now I will talk about the importance of proper aseptic method in microbiology. It protects the integrity of your culture. The aseptic method ensures that you only have your desired bacteria colony. It eliminates other microorganisms, communities, and prevents cross contamination and provides efficient result. It also provides safety to microbiologists, protecting them from harmful microorganisms. So now we will talk about the staining bacteria or fungi. The sterilize the slide with isopropyl alcohol will make sure it is clean, and also it should be cloudy. Add tiny drop of water. Now add tiny drop of water. So get your petri dish containing bacteria and transfer desired bacteria to your slide. The inoculating loop should be sterilized at 45 degrees Celsius over the alcohol lamp. Now check temperature of the inoculating loop, as it shouldn't be too hot. To check for the temperature, you have to tap at the end of the petri dish. And it is sizzles. It means that it's still hot. So you have to wait till it cools down. Hence, once the step is done, tap on the desired colony, and then transfer it to the slide by mixing it with the tiny drop of water on the slide. Then allow slide to dry. Once it's dry, to just have to keep it over the lamp. After that, you can rest the propyl staining method. So the first step is to add crystal violet dye to your slide. You have to completely wash your slide with crystal violet and wait for 30 seconds, 1 to 1 minute, for the bacteria to properly absorb the crystal violet dye. After 30 seconds, wash your slide with distilled water before adding iodine. Then add iodine and wait for 30 seconds to wait for bacteria to absorb it. Then wash it with distilled water, adding or washing slide with ethyl alcohol. Wait for 30 seconds to wait for bacteria to absorb it. Wash it with distilled water. Add saffron, wait for 30 seconds to 1 minute. Then wash slide with distilled water. Then dry the slide and view it under microscope. Now the staining method for fungi. Add a tiny drop of methyl blue to your sterilized slide. The staining method is quite complicated in fungi, in case of fungi, as it contains a spore and hyphae. Get a strip of tape and give desired colony a light tap and transfer to slide containing methyl blue. Get a cover slip which is clean and not cloudy. Cover your slide and then view it under microscope. So now to get your hyphae, you need inoculating liner and then sterilize and check the temperature is right. Pick desired colony of fungi and give it a light tap to get the hyphae. Transfer it to slide containing methyl blue and cover with cover step slip and view it under microscope. The precautions to take care while doing the staining process of fungi. First, make sure all the fans are off so that the spores don't blow in the lab. So thank you ma'am. This is in my video on how to transfer bacteria on petri dish, importance of aseptic techniques in the microbiology lab and staining method of fungi and bacteria. So thank you ma'am, thank you everyone. Salamat po.